Let's start with the name of Allah who is most beneficent and the most merciful. Assalamu alaikum everyone. I'm Rida Athar from the Department of Life Sciences. I am doing microbiology and I'm from 5th semester. So the topic of my today's presentation is vaccine. My instructor name is Sir Athar Hussain. Here are my group members: Seher Sajid, Zirwa Rahat, Munazza Shafiq and Marwa Abbas. Here is the overview of today's presentation. I'll tell you the introduction of vaccine, its history, how it works, types of vaccines, and discuss risk and benefits associated to vaccines. So let's start with the introduction. Before starting the actual topic, I'm giving you a brief introduction of what immunization is. Immunization is a process by which a person becomes protected against a disease through vaccination. Or you can say that it is a process by which a person becomes protected against a disease through vaccination. This term is often used interchangeably with vaccination or inoculum. There are two types of immunization: the active immunization and the passive immunization. Active immunization is when you give a vaccine and your immune system kicks into higher gear and sets up a series of reactions into your body to trick your immune system, thinking that you have actually had that disease. In active immunization, usually killed live or attenuated pathogenic organisms are used. Active immunization is long-term and play an important role in your immune system. While passive immunization is when you get those preformed antibodies. So the transfer of preformed antibodies, it is usually short term and it plays no role in your immune system. Vaccine. A vaccine is a substance or a biological preparation that provides active acquired immunity to a particular infectious disease. A vaccine typically contains an agent that resembles to a disease-causing microorganism and is often made from weakened or killed forms of microbes, its toxin, or one of its surface protein. Vaccine is a type of active immunization that it plays important role in your immune system, and they are usually long-term. So I'm clearing the difference between vaccination or immunization. Vaccination is the use of vaccine to give you immunity to a disease, while immunization is the process that happens in your body. Usually that is due to vaccination, but it can come from other exposure of a pathogen too. History of vaccine. So Edward Jenner is considered the founder of vaccinology in the West in 1796 after he inoculated a 13-year-old boy the vaccinia virus, cowpox, and demonstrated immunity to a smallpox. In 1798, the first smallpox vaccine was developed. Second important name is Louis Pasteur. Louis Pasteur performed experiments, spearheaded the development of live attenuated cholera vaccine, and inactivated anthrax vaccine in human. It is usually done in 1879 and 1908 respectively. Here in the diagram, you can see that in 1988, there are so many countries, like there are a lot of countries that can have never eliminated polio virus, and these are colored in pink, and the white area shows that these countries have eliminated polio. While you can see in 2014, with the help of vaccine, the world has eliminated polio virus, while there are just a few countries left that never have been eliminated polio. So let's get to the mechanism of vaccine. It is an ideal scenario whenever a vaccine is first administered, it is phagocytized by an antigen representing cell. So recent studies have suggested that it is particularly important that the vaccine can be taken up by the dendritic cell. So we can say that dendritic cells play an important role in the in producing T cells that play a role in immunization that will give your body immunity. So dendritic cells play an important role because they activate the T cells, which later become helper T cells. From there, the active cells goes on and become mature B cells. So these activated B cells divided into two types. The first one is antibody producing plasma cells, and the most importantly, memory B cells. Memory B cells play important role in the long term immunization. Memory T cells are also established. However, they usually have a shorter lifetime than memory B cells and thus play a minor role in long-term immunity. Usually, there are no cytotoxic T cells formed whenever the body responds to a vaccine. Now I'm discussing the types of vaccine. First of all, I'm going to explain inactivated vaccines. For these vaccines, a specific type of virus or bacteria is killed with heat or chemical, and its dead cells are introduced into the body. Even though the pathogen is still dead, but it will cause your immune system to learn from its antigens how to fight a live version of it in the future. These type of vaccines are usually specific for polio, hepatitis A, and rabies. Let's discuss the advantages of inactivated vaccines. These vaccines can be freeze-dried and easily stored. Easily stored because there is no risk of killing the pathogen as there is with live attenuated vaccines. These vaccines are safer to use and no risk of the virus or bacteria to mutating back into its disease-causing form. The disadvantage of these vaccines is that, as we know that the virus or bacteria is dead, so it is not as accurate a stimulation of the real thing as a live attenuated virus. Therefore, it will take several doses, and the process of vaccination and forming immunity in your body is usually slow, and we need booster shots to train the body to defend itself. Now, the second type is subunit and or conjugate vaccine. For some diseases, scientists are able to isolate a specific protein or a carbohydrate from the pathogenic organism, and then they will inject it into your body. This can help to train your immune system to react without provoking the sickness. In conjugated vaccine, polysaccharides can disguise a bacterium antigen so that the mature immune system do not recognize them. So subunit and conjugated vaccines are specific for hepatitis B virus, hepatitis B disease, influenza, Haemophilus influenza type B. So here is the advantages and disadvantages of conjugated vaccine. With these vaccines, the chance of an adverse reaction in the patient is usually much lower because we do not inject the whole pathogenic organism, we inject a part of it. Disadvantages. The main disadvantage of this vaccine is that the best antigen and the pathogen for training the immune system is usually difficult to identify, and then separating them is not always possible. So these type of vaccine will take a lot of time, and only a certain type of vaccine can produce in this way. These are the most important type of vaccine, DNA vaccines. DNA vaccines are still in experimental stages. DNA vaccine would dispense with all unnecessary parts of the bacterium or viruses instead of contain just an injection of a few parts of the pathogen's DNA. As a result of these vaccines would be very efficient immune system trainers. They are also cheaper and are easy to use. These type of vaccines are specific for influenza and herpes viruses and are still in human testing phase. So here is the diagram which explain how DNA vaccines can work. Here you can see in the step 1, a DNA vaccine consists of a plasmid produced in bacterium that encode the protein of interest in the presence of mammalian promoter. It is placed in a way that it reaches the cell nucleus, and enabling the transcription and translation in the transfected human cell. In the first step, while in the second step, that is 2A, after the plasmid uptake in vivo, the encoded protein is expressed into the host cell. The vaccine antigen can be then presented in antigen representing cells, that is APC, such as the dendritic cell through the major histocompatibility complex, that is MHC. Pathways can be presented to activate T cell, CD8 plus T cell immunity is predominantly activated by endogenously expressed antigen presented on MHC class 1 molecule, this one. After that, the active CDT T cells stimulates the release of cytokines and tumor necrosis that inhibited viral replication and increased expression of MHC1 a molecule. Therefore, macrophages here, the macrophages are also activated to support the cell-mediated response immune system. While you can see in the third step here, the CDT4 plus T cells activation is triggered by MHC2 class from the APC. In the case of protein are secreted, these are triggered, targets are recognized by the B cell receptors in naive B cells, which also use MH2 to get activated. In this immune pathway, activated B cells will also produce through different classes of antibodies and protect against the disease. So we can say that there are two types of immune response will occur. The first one is a cellular immune response, that is the effective immune response, and the second one is hormonal response. So these two immune responses are due to the DNA vaccine. Recombinant vaccines, recombinant vector vaccines. These experimental vaccines are similar to DNA vaccines in that they introduce DNA from a harmful pathogen into the body and triggering the immune system to produce antigen and train itself to identify and combat the disease. So the main difference between DNA vaccine and recombinant vector vaccine is that the recombinant vaccines use attenuated or weakened viruses or bacterium as a ride or vector for the DNA. These type of vaccines are usually specific for HIV, rabies, measles, and are still currently being developed. So here is the diagram. I'm going to explain how recombinant vector viruses can work. As you can see here, there is hepatitis B virus. Okay. HB antigen producing gene is extracted from the DNA of it. And here you can see the bacterium cell is present. It contains a plasmid. Plasmid is a circular DNA. We extract the plasmid and cut the plasmid with the help of restriction enzyme. Then we attach the gene of interest with our desired plasmid, and it will form a recombinant DNA. After that, this recombinant DNA introduced into the yeast cell, and this will make a recombinant yeast cell. After that, this yeast cell is placed into the fermentation tank where these yeast cells can increase in number and producing HB antigen in fermentation tank. Here you can see antigens are produced, and after that, extraction and purification of HB vaccine can occur. In this slide, I will tell you what are the risks of vaccination. There are usually very rare and very mild side effects, and that can be result in vaccination. These side effects completely depend on the immunization you got and how your specific body will react to it. Mild side effect. As the vaccines enter into your body, it is pretending to be the infection. You may get some of the symptoms of that disease, such as a cold, fever, or cough. Rare side effects. There are very rare chance that you get a severe side effect that come from vaccines, such as a high fever, rashes, or neurological episodes, but these are very rare. Allergies. Rarely, individual will develop allergic reaction to in response to vaccination and can have reaction in their shots. Allergic reaction can be very dangerous, but again, I'm telling you that this is a very rare case. Benefits of vaccine. So, vaccine play an uh, important and extremely effective in controlling and eliminating the dangerous disease. We can eliminate smallpox have been completely eliminated by the help of vaccination. Herd immunity. When more people have vaccination, it makes everyone less likely to get the disease that is vaccinated against. This control of disease is called as herd immunity and benefits the entire community. I hope you like the presentation. Thank you for listening.